One day, way back in 8000 BC, a nomadic tribe in southern Mexico stumbled upon teosinte, a wild grass with skinny ears and kernels wrapped inside a stone hard casing. With features that are so peculiar, it's hard to believe that teosinte marks the start of a 1,000-year long journey for an amazing crop. While they gathered for food, they noticed that there were slight differences in each teosinte plant. One had kernels that were at least partially exposed. One exhibited ears that held together better. One contains more rows of kernels. The teosinte seeds exhibiting those traits were grown together, resulting with a teosinte plant possessing all these traits, a process which we now call selective breeding. This process was patiently repeated for thousands of years until they were able to transform a grass with many inconvenient, unwanted features into a high-yielding, easily harvested crop: corn. Today, thanks to the advancements in technology, scientists have created a new, faster way of breeding, one of which is called embryo rescue. Embryo rescue is an in vitro culture technique that assists in development of plant embryos to become viable plants. Immature embryos are isolated from developing kernels 14 to 17 days after pollination. These embryos are then grown in nutrient media. Mineral salts, sucrose, ammonium salt, and various amino acids are essential requirements in the culture of medium for proper growth and differentiation of embryos. Initial dark incubation for 4 days of embryo culture is essential. In the following days, these can be grown to maturity under light conditions. Within 6 to 10 days, germination occurs and the plantlets start to grow, after which are transplanted into soil. This process hastens the development of the corn embryo. The growth and development of corn does not stop there. The embryo then differentiates into a shoot apical meristem, which is responsible for the growth of the shoot system, and a root apical meristem, which is responsible for the growth of the root system. Roots and shoots are composed of 3 types of tissues, namely dermal, ground, and vascular tissues. Dermal tissues form the outer protective layer of the plant. These tissues are made up of epidermal cells and specialized cells such as guard cells and root hairs. On the other hand, ground tissues consist of parenchyma cells that function in storage, photosynthesis, and secretion. Lastly, vascular tissues consist of xylem, which conducts water and dissolves minerals, and phloem, which conducts food such as sugars throughout the whole plant. Those are cells and tissues that make up the amazing and delicious food crop. As we learned how corn develops and grows, we should also learn more about its uses. We are all familiar with corn as a star of dining tables, but this amazing plant is also a star of genetic studies. Corn was noticed early on because of its large and easily interpreted meiotic chromosomes, which are used as visual aids for studying genes and their mutations. In fact, mutants affecting all stages of meiosis, ranging from the initiation to the regulation of chromosome pairing and control of the second division, have been carefully described, and many of the casual genes have been cloned. Maize is highly utilized in a lot of genetic studies, and one interesting example is forward genetics. There are many methods existing for forward genetic screens, including chemical, radiation, and transposon-based mutagenesis. Ethyl methane sulfonate, or EMS, is the most efficient and widely used chemical mutagen. It alkylates guanine, causing a G to A transition point mutation that results in both dominant and recessive mutants. First-generation offsprings are then self-crossed to reveal recessive mutations in the second generation. EMS-based mutagenesis has yielded many important maize mutants that have advanced the understanding of plant architecture, meiosis, kernel and embryo development, photosynthesis, and disease symptoms. Loss of dominant kernel phenotypes was one of the first indications that ionizing radiation can lead to the gross phenotypic alteration and embryo lethality. These phenotypes were the result of large-scale chromosomal aberrations, such as deletion, breaks, translocation, and initiation of breakage fusion cycle that led to ring chromosomes, dicentric, and other unstable chromosome features. Ionizing radiation has also been used to generate mutants in somatic tissues. Radiation of somatic cells at specific developmental times allows researchers to study mutations that would normally be lethal. Truly, maize is a beautiful plant with extraordinary wealth of genetic tools and genetic uses. The story of corn is just one example of how God created our world with an abundance of natural beauty and wonder. But aside from that, He also created us, human beings, and gifted us the knowledge and capability to utilize and improve the natural world. It now rests in our hands to make sure that the knowledge we gain from the world will be used for the benefit of it. Like how the Native Americans created a better world when they transformed a useless wild grass into an amazing plant.